Dr. Gupta, and she's from Department of Zoology. She will be speaking on heterogeneity in the fat body tissue revealed using single cell RNA sequencing. Over to you.
Audience for sticking around. So today I'll be speaking about the role of the fat body tissue in immunity and reproduction, which we are addressing using single cell sequencing as the title suggests. So if we look at the interactions between host and pathogens, it's like an arms race where if a host successfully defends itself against a pathogen, then the pathogen can quickly counter-evolve, resulting in this kind of an arms race, which is a constant tug of war. And I think Professor Gupta in the morning very brilliantly explained how quickly bacteria can evolve resistance against multiple defense targets. So now, because of this threat of the pathogens that we have, the immune system that we have is multi-layered. So there are different components to the immune response, ranging from physical barriers, which prevents the entry of the pathogens, to specialized proteins and cells, which can target these pathogens and kill these pathogens. Now, this immunity, howsoever important it is, can be affected by several different factors. It could be our own physiology. So how old are we? How active are we? How fit are we? Similarly, our genetics could also affect our immune response, the environment we live in, and given we are in a nutraceutical conference, the kind of nutrition that we consume can also affect our immune response. So for this presentation, I am specifically focusing on the effect of reproductive status on the immune response. And if we look at the examples in the literature that is there, more often we see that the reproductive physiology often negatively affects immunity. So ranging from insects to birds to mammals, the reproductively active organisms often have poor immune response. I am an evolutionary biologist, so in our terminology, these negative relationships that we see between the traits, we refer to them as trade-offs, which essentially means that there are often constraints, which could be physiological limitations. It could as well be the availability of the resources, which could be limited, as a result of which, if investment goes up in one trait, so in this case, let's say reproduction, it can come at the cost of another trait. So in this case, that would be your immunity. Now we see a lot of the examples, and I already showed you the diversity across which we see this, but we do not really understand that why reproduction and immunity are negatively correlated. So this was the question that we tried to address. The organism that we used was Drosophila melanogaster. So I think it's a wonderful organism. We can measure all the different traits in the organism, be it immunity or reproduction. In the lab, we also see that reproduction has a negative effect on the immune response in Drosophila. So if mated and virgin Drosophila females are infected with a gram-negative bacterium, Providencia redteri, so the way infections are done, so a thin needle is dipped in bacterial suspension and the flies are infected with this bacteria. So when mated and virgin females are infected, so on the x-axis is hours post-infection, on the y-axis is survival post-infection. What we see is that mated females have lower immunocompetence, or they die more because of this infection with Providencia compared to the virgins. So we asked that why do mated females have poor immune response? For that, we focused on the fat body tissue in insects. So it's one of the major organs in the insects, and it controls multiple functions. For our interest, it controls both reproduction and immunity. So we wanted to understand that how is the fat body tissue controlling so many functions? Does the fat body tissue consist of multiple subpopulations, where each population has a specific function, or each cell perform these functions, and as a result of which they are constrained when they have to control multiple functions? So the hypothesis was such that, let's say under the basal conditions, we have a specific set of subpopulations when the flies are mated, different subpopulations respond differently. Now their gene expression can change, their cell numbers can change, or the new cell populations may arise due to this change in the physiology. On the other hand, upon infection, there are different subsets of cells which are affected. And when there is a dual stimulus in the case of, let's say, mating and infection, these cell populations are then constrained. And as a result of which, we see a negative correlation between reproduction and immunity. So in the experiment, we collected our samples 24 hours post-mating, and the infected samples were collected 6 hours post-infection. So for this, we took 4 Drosophila, 4 treatment groups, basically virgin uninfected, virgin infected, mated uninfected, and mated infected. We dissected their fat body tissues, and we did something which is called as single cell sequencing. So essentially in this case, a single cell is entrapped in a droplet which also contains all the reagents to prepare a cDNA molecule. So all the mRNA molecules are converted into cDNA within this droplet. A barcode is added on that cell basically, and that's how then by computational pipelines we can understand that what gene expression came from which particular cell. So this is what we did. Now coming to the results. So here it's a 2D projection of a multi-scale data basically. Each dot is a single cell. Its proximity to the other dot or other cell is dictated by the overlap in their gene expression. Greater the overlap, greater the proximity of these two cells would be. So we were able to identify the fat body subpopulations within our data set. When we were doing dissections, the fat body tissue is extremely difficult to kind of take it out essentially. So there were other tissues which were attached with this fat body, and by using the predefined markers, we were able to identify those cell populations and kind of focus just on the fat body cells. So this is the same graph color coded in our treatment groups. And then we looked at the gene expression across those treatments. What we saw was following, that in the virgin samples, so virgin uninfected are here, virgin infected are here, and we saw that both of them have higher ribosome biogenesis. In addition to that, virgin infected females also showed lower ER stress. On the other hand, the mated uninfected females had lower ribosome biogenesis, they had higher metabolism. And mated infected samples showed lower ribosome biogenesis and higher ER stress. So we wanted to understand what does this really mean for the biology of the tissue. So Ananya in the previous talk talked a lot about protein folding. So when ER stress happens, it's the proteins which are not properly folded. And as a result of which, the misfolded proteins begin to accumulate in your endoplasmic reticulum membranes, because of which these membranes dilate or they swell up. So we looked at that using electron microscopy. So the fat body cells from the virgin infected showed really constricted or the natural ER membranes. On the other hand, in the mated infected samples, these ER membranes were highly dilated. We also saw changes in the ribosome biogenesis. So it basically included genes such as which produces different subunits of the ribosomes and rRNA genes, as well as translation initiation factors, suggesting that if ribosome biogenesis is affected, in the downstream it will affect the protein synthesis. So we next asked if the reduced ribosome biogenesis results in reduced protein synthesis in the mated infected. So to understand that, we did some Western blotting using puromycin. So it's an amino acid tRNA analog which gets incorporated into nascent polypeptides. So higher the incorporation of puromycin would indicate higher rate of translation. And that's exactly what we are seeing here. And when we did some quantitation on these, we could show that virgin infected females had significantly higher protein synthesis compared to the mated infected females. So the model that we worked out was the following, that if infected virgin females can immediately synthesize immune-related proteins upon infection, they have better survivorship. On the other hand,
Dr. Gupta,
mated infected...
Excuse me, can I ask you to wind up in 5 minutes?
Sure. In 5 minutes.
Yes.
So mated infected females had lower ribosome biogenesis and higher ER stress, because of which they are not able to start the protein synthesis, resulting in poor survivorship. So if we change the protein synthesis during reproduction, then can we really go for better survivorship? So skipping these schematics, if people are interested, I can talk about it later. We basically treated flies with cycloheximide during the time of their mating, and we allowed the flies to recover from this cycloheximide effect and then infected them. When we look at their survivorship, we showed that the cycloheximide treated mated infected females survived significantly better compared to the mated infected females. When we look at the protein production, again cycloheximide mated infected females had significantly higher protein production compared to the mated infected. We also looked at the production of immune peptides by using some Mardi-Top. So here it's just a spectra of Mardi-Top of antimicrobial peptides. And we again showed that the number of peptides, immune peptides that are produced by virgin infected and cycloheximide treated flies is significantly better compared to the mated infected females, suggesting that this is in some sense a plastic response. So again, to put everything in perspective, it's possible that by providing amino acids or changing the diet, we can again maybe influence the translational capacity. So to conclude, mated females upon infection suffer from suppressed ribosome biogenesis. Consequently, mated infected females synthesize less proteins when they are infected, and translation regulation at the time of mating can later direct translation machinery towards infection. So essentially, using fat body for multiple processes constrains its translation capacity. And with this, so this is my postdoctoral work, and I would like to thank my mentor, Professor Brian Lazaro, the scientist in the lab, Dr. Ashley, and my undergrad, Nick, which helped me with a lot of gene sorting. And with this, I take any questions. Thank you so much.
Thank you, Dr. Very wonderful talk. You have added a new perspective to Peter Medawar's theory. So,
Kalpana, there is a it's just a suggestion, if you had taken microbiome also into consideration in the virgin and mated females, probably linked it with the changes in the fatty acid tissue.
Definitely, sir. So with these setup, we haven't shown, but I think
because everything is now percolating to.
But there is evidence that there isn't a huge shift between virgins and mated in the terms of microbiome. So I guess in this context, but definitely.
So what is the, this is not with respect to the way you are looking at, but otherwise I'm asking, like if say for example there are these infected females, what is their like reproductive output or capacity? Do you see certain kind of change there?
It's highly variable from pathogen to pathogen. For this particular pathogen, if we compare, if we match for the window, because these flies would also begin to die. So if we consider their, as long as the infected females are living and we compare that to the uninfected females, the reproductive output do not change.
It doesn't change,
at least with this case.
Okay.
There are certain bacteria where actually infected females lay more eggs, because we call it as terminal investment, so they are trying to reproduce or produce as much progeny as possible before they die.
Okay.
So,
and another thing which I wanted to ask, just one second. So another thing which I wanted to ask you is, you did single cell analysis, especially the RNA analysis. So did you also look into what are these different populations, the kinds of cells basically? Have you done that profiling as well?
Yes, theoretically yes. So I have looked at all the markers and what those markers would mean. I didn't have a chance to kind of then basically, I mean, the next course of action typically would be to knock down those markers and see how it's affecting those populations. We haven't gone that far, or we haven't really physically looked at those on the tissue. But in terms of markers and the putative function that the cell population might have, that much we have already done, and that's also included in the paper.
Yes. Okay.
I have two questions. One is regarding fat body. This fat body does resembles human adipose tissues.
There is similarity in terms of fat storage, yes.
Okay, and also like second question related to like after infection, like usually like after in mating, protein production will be high,
Yes.
but according to your presentation, it is low. What is the reason?
So I'll come to that. So that's where it was a lot more confusing for us to begin with. So their ribosome, when I say their ribosome biogenesis is reduced, it's the gene expression which is reduced. But when I look at the protein synthesis, so their protein synthesis in mated uninfected is similar to the virgin uninfected. So we think that this is what the basal state of protein expression is. That is probably where the homeostasis kind of comes into the picture, and that's the basal level. The fact that their gene expression, or they don't already have mRNAs to synthesize new ribosomes, is where we think that the mated infected females are not able to make new proteins.
But you told like they lay more eggs in some bacteria.
With some other bacteria, so we don't know how.
But like even for laying more eggs also they need more protein synthesis.
So a lot of protein synthesis and yolk production happens in the ovaries as well, and we don't know whether how the translation is affected in the ovaries. The other thing, the translation inhibition that we are seeing is also specific to this pathogen as of now. We are one of my projects is to kind of test how general this phenomena is, and then I think I'll be able to better answer your question.
Since protein synthesis machinery was disrupted, that's why translation was.
Yes. Ribosome biogenesis is disrupted, and that's why.
It's a consequence of that.
Yes. Yes.
Thank you so much, Dr. Vedika. So I think I interrupted you guys so that we could have a lot of questions. Thank you so much for a brilliant presentation. So we wrap up this session. Very burning one. Thank you.
Thank you, Dr. Gupta. Now I request Professor Rubla to facilitate.
Yes, sir.
Thank you, sir.
So this session ends now, and any announcement, please?
Now I request Professor Anju Srivastava to facilitate the chair.
Sir?
कौन किसको दे रहा है?
नाम लिखा नहीं तो मैं देख